Hello guys, welcome to my YouTube channel. So in this video, we are going to learn about the electron transport chain. So this is the part of the body or the process that occurs in the body that enables the body to generate energy in form of ATP. The energy you have for movement, for doing all the other activities is generated from the electron transport chain. So today, we are going to have an overview, or we are just going to have knowledge about what happens inside the mitochondria that leads to the generation of that energy that we use for the various processes that we undertake. Now, let's start from here. Krebs cycle. Krebs cycle occurs generating NADH and FADH2. It also generated some amount of ATP, but you're not going to account for that. Also, glycolysis produce some NADH. So these are the precursors, these are the raw materials that you're going to take into the electron transport chain so that you can generate ATP through a series of complexes here. Now let's get started. This is the first complex, or you can call it NADH dehydrogenase, or you can just call it complex 1. Right? This one here is coenzyme Q. Coenzyme Q. In some books you can see it written 10. Coenzyme Q10. Right? Then this one is called succinate dehydrogenase, or you can just call it coenzyme, complex 2. You can just call it complex 2 or succinate dehydrogenase. Then this is another coenzyme Q. This is another coenzyme Q10. Then this is complex 3. Or it has a great long name. You cannot call it coenzyme Q cytochrome B oxyreductase. That is a very long name, but you can just call it complex 3. Then this is called cytochrome C. Cytochrome C. Then this one is called cytochrome oxidase or complex 4. Cytochrome oxidase. Right? Yeah. So those are the complexes we have in the electron transport chain. And you know, electron transport chain or the generation of ATP occurs inside the cytoplasm. So inside the cytoplasm, specifically inside the mitochondria. So these are mitochondrial organelles inside the cytoplasm of the cell. Right. So this is the inner membrane, this is the outer membrane. Here is the matrix. So the complexes are located between the inner membrane and the outer membrane. Now let's see. We are going to feed these precursors, the NADH, FADH2, inside the electron transport chain. So let's see what's going to happen. Let's start with NADH. So NADH is going to come, NADH, and it's going to drop the electrons. So remember, NADH has hydride electrons. Hydride, just as I told you, hydride simply means a proton plus 2 electrons. So this is the hydride. So it's going to get the electrons inside the complex 1, and the electrons are going to be taken up by a molecule called fleaving mononucleotide, and this is going to be oxidized to NAD+. So fleaving mononucleotide is going to take the electrons and forms fleaving mononucleotide with the electrons H2. Then this is going to be oxidized to NAD+. Then this coenzyme Q. So fleaving mononucleotide doesn't have the electrons, that's not the high energy condition. So it's going to take the electrons, so pass the electrons to coenzyme Q. So here is the coenzyme Q. Coenzyme Q is going to take the electrons, right? So this can pass the electrons and it be converted to fleaving mononucleotide. Then coenzyme Q is going to take the electrons, then be converted to QH2. Yes. So remember, one thing you must take into consideration. When we are passing the electrons from complex 1 to complex 2, there's something funny happens. Now remember, inside the cytoplasm, there are protons. Inside the matrix of the mitochondria, not the cytoplasm. Inside the matrix of the mitochondria, there are protons. So these protons are going to be pumped out of the matrix inside the mitochondrial or intermembrane space of the mitochondrium via this receptor there. They are going to be passed out. So they are passed whenever the electron are passed from complex 1 to coenzyme Q. It creates some energy that's going to pass the protons out, or going to force them into the intermembrane space. Right. Then coenzyme Q. Let's start from there a bit. So remember, complex 1 only takes NDH. It doesn't take FADH2. So let's go to complex 2. Complex 2 takes the FADH2. So we're going to have FADH2. It's going to be converted to FAD plus. It's going to be oxidized. Then the same thing is going to happen. The energy, the electrons are going to be taken up by fleaving mononucleotide and fleaving mononucleotide with electrons. But remember, complex 2 doesn't have the receptor for pumping the protons out of the cell. Mostly inside the cell, the concentration of protons is about 1 times 10 to negative 9 millimolar. So we want to see how we are going to create the concentration gradient so that we generate energy in form of ATP. So FADH2 is going to come to complex 2, not complex 1, and NADH cannot come to complex 2. So it's only the FADH that unloads these electrons to fleaving mononucleotide in complex 2. So we have fleaving mononucleotide. Then you have another coenzyme Q. So fleaving mononucleotide is again going to pass the electrons to coenzyme Q, and it is converted to fleaving mononucleotide, ready to take another electrons from another molecule of FADH2. Then coenzyme Q is going to take the electrons and converted to QH2. Coenzyme Q with electrons. Then coenzyme Q are going to combine their electrons. So this one is going to pass the electrons to that's coenzyme Q. Alright. Now let's see. Coenzyme Q is going to pass the electrons to complex 3. Remember, none of this particular complex has that high energy state. One those electrons. They are going to keep on passing them all over again. Now, so you have coenzyme Q to complex 3. Complex 3 has cytochrome. It has cytochrome B. It has cytochrome B, which is bonded to ion 3 and ion 2. So coenzyme Q is going to pass the electrons to ion 3 and is converted back to coenzyme Q. So remember, QH2 has electrons, Q doesn't have the electron. So it's going to pass the electron to ion 3. Then ion 3 doesn't want the electrons, it converts to ion 2. And remember, you see, here there is a receptor. So whenever this complex 3 is going to pass the electrons to cytochrome C, we are going to have the protons being pumped out of the matrix. So these are the protons. Now, ion 2, so this cytochrome C also have ion 3 and ion 2. So ion 2 is going to pass the electrons to ion 3 and is converted back to ion 3. Then ion 3 of cytochrome C takes the electrons and is converted to ion 2. But remember what I told you, whenever it passes the electrons from complex 3 to cytochrome C, it's going to generate some energy that is going to pump the protons from inside the matrix to the intermembrane space. So you have more of the protons are being pumped out, increasing the concentration of the protons inside the intermembrane space. Now, cytochrome C is again going to pass the electrons to complex 4. So, complex 4 has cytochrome A plus A3 and it's also bound to ion 3 and ion 2. Right. Now, let's see. Cytochrome C is going to pass the electrons to complex 4. Right? So, let's see. Ion 2 is going to pass the electrons to ion 3, and ion 3 take the electrons and is converted to ion 2. Right. Now, where else are we going? We have reached complex 4. So remember, the last electron acceptor in the electron transport chain is oxygen. So complex 4 doesn't have anywhere to unload the electrons apart from oxygen. So you have atomic oxygen here. Atomic oxygen is going to accept the electrons, right, and combine with the protons to form water. So it's going to form a water molecule. So that electron, oxygen is the last electron accepted in this particular process, because in this particular series of reactions. So it is the last electron accepted because none of them want the electron. So you see, it keeps on passing past the electron until we reach oxygen, right? Now, remember I told you whenever we are passing electrons from complex 4, remember it has a receptor. So it's also going to pump the protons out of the matrix into the intermembrane space. At the end, you realize that we have like almost 1 times 10 to negative 7 millimolar concentration of protons in the intermembrane spaces compared to this one. So there's going to be a concentration gradient that's going to be created due to high concentration of protons inside the intermembrane spaces compared to the matrix. And that's the main reason why we are going to generate ATP. We are going to use that mechanism to generate ATP. Now, let's go to complex 5, which is called complex 5 or ATP synthase sometimes. It's called ATP synthase. This is it. Now, ATP synthase is going to take advantage of the proton gradient created due to pumping in and out of the protons, of the electrons, the electrons. So, the electrons are here. The protons, not electrons. Electrons have been taken by atomic oxygen to form water. So, here the protons have been pumped into the intermembrane space. So, the protons are inside the intermembrane space. The protons are going to get in via a receptor. So remember this receptor, it has something, it has a protein molecule inside here called arginine 210. Let me write arginine 210. So arginine 210 is going to take electron from this side and pass it over to the other one, to the other opening. So it's going to take it from that opening, pass it over. So the more it happens that way, it's going to rotate. So when it rotates, it's going to rotate the ATP synthase. So we can just call this a rotor. And you can call this a rotor. So it's going to take the protons from inside the intermembrane space into the matrix of the mitochondria. So the more it does that, it's going to do this, it's going to rotate. So the more it rotates, it's going to rotate the motor or the ATP synthase. So this is going to lead to generation of some potential energy. So this potential energy is going to be taken up by the ATP synthase. So let me zoom out this one so that you can see what happens. These two protons. So here there is an organic phosphate, and here there is ADP. So whenever it rotates, it's going to take in some of the potential energy. So just does this form ATP, does this form ATP. So it's going to combine organic phosphate and ADP. So organic phosphate is going to be added to ADP to form ATP. That's what we wanted. So remember, the receptor here is going to take in protons from inside the intermembrane space into the matrix by the help of arginine 210 protein. As the arginine 210 carries the protons from inside, it's going to rotate. It take it and pass it over to the other opening. Take and pass it over to the other opening. Then as it does that, it's going to rotate the motor or the ATP synthase, which is going to lead to generation of some potential energy that's going to be used to add phosphate molecule into ADP to form ATP. Now, you will realize that when we break down one glucose molecule to do the process of glycolysis to create circle to generate NADH and FADH2, we are going to have close to 30 or 32 or up to 38 molecules of ATP depending on the number or the ratio you are going to use. Now, let me also show you something here. Remember glycolysis, we converted to glucose to pyruvate. We generated some NADH. And remember, we need NADH for the electron transport chain. So what are we going to do? We want to find ways in which we can get NADH inside the mitochondria so that can undergo the electron transport chain process. Now, so we have NADH. NADH is going to be converted to NAD+, going to be oxidized. And remember, inside the cytoplasm there is oxalic acetate. Right? So oxalic acetate is going to take the electrons and is going to be converted to malate. This is a 4-carbon molecule. This is a 4-carbon. It's going to be converted to malate. So malate has the ability to penetrate the mitochondrial membrane. So it's going to come into the mitochondria. Inside the mitochondrium, it's going to, so remember, it has the electrons, right? So it's going to deliver the electrons inside the mitochondria. So it's going to be, the electrons are going to be taken by NAD plus. Let me have this picture. NAD plus and converted to NADH. And the malate is going to be converted back to oxalic acetate. Let me abbreviate that way. And remember, these are what we needed here. So we already got the NADH generated from glycolysis into the electron transport chain, right? Yeah. Now, we need to get this oxalic acetate back into the cytoplasm so that we can continue taking the electrons from the glycolysis NADH into the electron transport chain inside the mitochondrium. So oxalic acetate, so you have the oxalic acetate, it's going to react with glutarate. Right. It's going to react with glutarate so as to generate alpha keto glutarate. And oxalic acetate is going to be converted to aspartate. Now, aspartate also has the ability to leave the cell, so aspartate is going to leave the cell. So you have the aspartate. Aspartate is again going to react inside the cytoplasm. So remember, when to generate oxalic acetate, so aspartate is going to react with alpha ketoglutarate again. It's going to react with alpha ketoglutarate. This is alpha. Correct. So what are we going to form? We are going to form glutamate. Alpha ketoglutarate is going to reduce to glutamate, and your aspartate is going to be converted to oxalic acetate. And that's what we have here. So this is the mechanism that the body uses to take in the NADH generated from glycolysis. Remember, glycolysis occurs inside the cytoplasm, and electron transport chain, the generation of ATP occurs inside the mitochondria. So we must find ways of getting these precursors of electron transport chain inside the mitochondria. So this is the mechanism the body uses to get the NADH with the electrons inside the mitochondria. So I think we've covered everything. So let's have a quick overview of what we've learned. So Krebs cycle generates NADH, FADH2. The NADH2, the NADH are going to be taken in to complex 1 and they're going to be oxidized to NAD+. It's going to leave the electrons which are going to be taken up by the fleaving mononucleotide. And the fleaving mononucleotide takes in the electrons and is converted to fleaving mononucleotide with electrons, FMNH2. Remember, it also has a receptor which are going to take in the protons from inside the matrix into the intermembrane space when it passes the electrons to coenzyme Q. Coenzyme Q takes the electrons converted to coenzyme Q with electrons, that is QH2. Then now we are going to come to FADH2. FADH2 unloads the electrons at complex 2. So the electrons are going to be taken again by fleaving mononucleotide. This is going to fleaving mononucleotide with the electrons. Then it's going to pass the electrons to coenzyme Q. Coenzyme Q takes the electrons, right, and combines. So the coenzyme Q from this side combines with coenzyme Q from this side, going to take the electrons to this other coenzyme Q. Then coenzyme Q passes the electrons to complex 3. Complex 3 has ion 3 and ion 2. It's called a cytochrome B. So it also has a set of passing in or pumping the protons from inside the matrix into the intermembrane space. Then protons are converted, electrons are passed over to cytochrome C. Ion 3 is going to take the electrons, pass to complex 4, going to take the electrons, converted to ion 2. Then the last electron acceptor, which is the oxygen, atomic oxygen, is going to accept the electrons combining the protons to form water. Right? Then you have the final one, which is ATP synthase. It's going to take advantage of the proton gradient, the gradient that has been created due to passing out of the protons. So the protons are going to be taken in by a receptor containing protein adenine 210. Protein adenine 210 is going to take the electron from this side, passing over to the other opening, passing over to the other opening. So as it does that, it's going to rotate. This rotation is going to pass over to the ATP synthase, and this will lead to generation of some potential energy. This energy is going to be used by the ATP synthase to add phosphate group into ADP. So whenever it takes in the potential energy, it does this way. It combines them to form ATP. So this is ADP plus an organic phosphate to form ATP. Right. So that's all about electron transport chain. And guys, if you felt that you've learned something from this video, keep in touch with me and make sure you subscribe to my YouTube channel for more videos. Thank you.